Hello students, welcome to the second video of control and coordination where we are going to continue with the movements that are shown by the plants that are Nastic movements. So without further ado, let's start. Here is my screen and we are going to discuss the following topics in this video. So we are going to start with the Nastic movements and its types, the photoperiodism, control and coordination. In control and coordination in humans, we are going to start with receptors, then the nerve cell. So nastic movements are the plant movements that occur in response to the environmental stimuli, but unlike the tropic movements, the direction of the response is not dependent on the direction of the stimulus. So in tropic movements, we saw that the growth movement is taking place in the direction of the stimulus. Here, there is no such limitation, okay? And the movement doesn't have to be in the direction of the stimulus. So these are some of the most spectacular plant movements. So how does it occur? How the nastic movement occurs in plants? This is a sensitive plant, which is the touch me not plant, Mimosa pudica. Everyone must have heard about it. Everyone must have seen it also. So this Mimosa pudica, when it is touched, the leaves fold up. Okay. This folding of leaves takes place in response to the touch stimulus due to the turgor pressure differences which takes place inside the cell. Okay. The turgor pressure is the pressure that develops in the cells when the water moves inside, the water is absorbed by the cells. So this is the pressure that is applied by the cells. It is applied by the cells on the water. Okay. So this is due to the turgor pressure differences in the cells. Okay, let's move on to the plant hormones. Plant hormones are also known as phytohormones, and these are the organic substances which regulate plant growth and development. Some of the plant hormones are auxins, gibberellins, abscisic acid, cytokinins, and ethylene. And these are the plant hormones which are found inside the plants. Rest of them, if they are applied to the plants, they are not natural. Okay, they are chemically synthesized, and they are one or the other variety of these hormones only. Okay, so let's start with the first hormone, that is auxin. Okay. In the previous video, I also explained the importance of auxin in phototropism, the function of auxin in phototropism. So this is the terminal bud where the auxin is synthesized and it moves towards the part which is non-illuminated. Okay. It causes the growth of the illuminated part towards the light, which is the terminal bud, but it gets diffused to the non-illuminated part where it causes the elongation of the cells. So this is the non-illuminated region, and this is the illuminated region which is exposed to the light, and it eradicates the auxins on the sunny side, which is written here, causing the cells on the shady side grown longer, and this makes the bending of the tip towards the sun. Okay. This experiment was actually performed by Boyson and Jensen. Okay, uh, they further the experiment of Darwin and Darwin, who explained the significance of auxin in the bending of the root tip, the coleoptile, towards the light source. Okay, so they excised tip the coleoptile tip. Okay, they placed it on an agar block, and they observed that auxin diffuses into the agar block. This agar block was then put on another excised plant. Okay, as you can see here in the control experiment where agar block is not there, the auxin has no effect here. Okay, and where the tip has an agar block, you can see the curvature is shown. Okay, this curvature is shown because the auxin, it can diffuse through the agar block into the rest of the plant. So this is the experiment to explain the significance of auxin and also to confirm it. These are the functions of the auxin. It shows apical dominance. Apical dominance means the apical buds will grow. Okay, and the lateral buds, the growth of the lateral buds is stopped. It prevents the growth of the lateral buds, but it promotes the growth of the apical buds, which is known as apical dominance. Okay, next is cell division and elongation. It causes cell division and elongation, and with the help of that, it helps in root initiation and development. This helps in growth of the plant. It causes phototropism and geotropism. As you can see, geotropism in the roots and phototropism in the shoots. It also promotes flowering. It helps in the flower development and it also helps in fruit development. These are some of the important functions of the first ever plant growth regulator that was discovered, known as auxin. Next is gibberellin. Gibberellin was actually discovered by Kurosawa. He was a Japanese plant pathologist. He was working in the rice fields, and while working in the rice fields, he saw that some of the rice seedlings, they grew taller than the others, and he saw that they got infected with a fungus, Gibberella fujikori. So this disease was known as Bakane disease. The disease that was caused by this fungus. Bakane in Japanese means foolish. So the rice seedlings that grew foolishly tall, they ultimately resulted in the death of few plants. That's why he called it Bakane disease, or the foolish rice seedling disease. Foolish rice seedling disease. Okay. Then later on, Yabuta and Hayashi, they isolated the crystalline sample of this chemical and they called it gibberellins. Gibberellins are the compounds which have a gibbane skeleton. Okay, and I have shown the gibbane skeleton here in the picture. There are more than 100 gibberellins found in that have been discovered. Okay, out of these, 125 have been isolated from this fungus. Okay, Gibberella fujikori, and 51 they have been isolated from the plants. So some of the functions of gibberellins are stem elongation, bolting. Bolting is basically the growth of the internode just before the reproduction. This is the growth of the internode in the plants. Okay, so in the rosette plants, what happens is there is profuse leaf development on the branches, on the nodes, but the length of the internode doesn't grow. The length of the internode remains the same. But just before the flowering begins, okay, the internode grows about it grows so long that the length of the plant, it becomes 5 to 6 meters. Uh, the the plant grows 5 to 6 meters tall, taller than the previous height. Okay, this process is known as bolting, where the internode elongation takes place just before the reproduction. Now, there are some long day photoperiod requirement for the rosette plants for the internode elongation, and cold temperature requirements are also there. But if the rosette plants, they are exposed to gibberellin, they are treated with gibberellin, then bolting can take place even before when the rosette plant leaf development takes place in the rosette plants. So it doesn't happen just before the reproduction, it can happen when the rosette plants start developing. Okay, next is seed germination. Then there is breaking of seed dormancy. Seed dormancy means the seed from being inactive, it will become active. Okay, and parthenocarpy is the development of fruits from unfertilized ovary. Okay, so with this, let's move on to this picture. Okay, where there is a mutant plant which remained dwarf, and when it was sprayed with dilute gibberellin solution, you can see the size of the plant. Okay, it induced fruit development, induced the development of shoots also, and you can compare the size of the plants basically. In the next picture, you can see there is a mutant plant which does not make any auxin. Okay, but when it was supplied with auxin, the auxin made it grow. Okay, as you know, auxin causes cell division, it causes cell elongation also. It also causes root initiation and shoot initiation, which helps in the development of plant. Okay, let's move on to the next hormone, that is cytokinin. Cytokinin, it was isolated by Miller from the herring sperm DNA. Okay, and this substance that was isolated, it was actually called kinin because it showed cell division activity. It showed cell division. Okay, later on, all the substances which show cell division, it was known as, they were called as cytokinin, because they had kinin like functions. Okay, in this picture, some of the functions of cytokinin are listed, like it helps in promoting cell division, it helps in organogenesis process, means the development of organs like shoot, roots, flowers, fruits, etc. Okay, this process is also known as morphogenesis. The third function is nodulation. Nodulation, as you know, is an important process of nitrogen fixation in legume plants, where the nitrogenous bacteria help in nitrogen fixation, and they live in the nodules of these uh, roots of these plants. It helps in delaying senescence also. Senescence is the old age. Okay, next is, it helps in sink source relationships. Okay, sink is uh, sink is the region where basically a particular substance is transported, and that particular substance shows its action. Okay, a response is given, where the response is given. Source is the uh, region where that particular chemical is synthesized. Okay, so it helps in sourcing's relationship also. Next is light mediated development. Then meristem function also. Meristematic cells are those cells which are actively dividing, and they are found in meristematic tissues. It also helps in nutrient response, apical dominance, which is the growth of apical buds, vascular development, and pathogen interactions. Okay, so this is the story of cytokinin. Let's move on to the next hormone, which is ethylene. Ethylene is the only gaseous hormone, and it is usually present in, uh, the smoke that comes out from the industrial gases. And this is the structure of ethylene. So this is CH2, CH2. Okay. The most important function of ethylene is in fruit ripening. It helps in ripening of the fruit. So if there is a raw fruit and ethylene is supplied to it, the indole acetic acid levels can uh, they will increase, okay, which will cause the ripening of the fruit by activating the ethylene receptors. Okay, so these are some of the functions: stem and root growth and differentiation, breaking dormancy, fruit ripening, flower and leaf senescence. Okay, and the next hormone is abscisic acid. It is also known as dormin. Okay, it is a stress hormone because it helps in promoting seed dormancy. It makes the seed dormant. Okay, it also helps in shoot development. It also helps in stomatal closure. It also helps in maintaining the root architecture. More functions we will discuss on the next slide. But Kahns and Adicott, they saw that the cotton leaves, the cotton balls, they broke due to a substance known as, they isolated the substance and they named it abscessin. Okay, abscessin 2, basically. Okay, then they later called it abscisic acid, and they found out that due to the presence of this substance, the cotton balls broke down. These are the functions of ABA in shoot and in the root. It helps in stomatal closure. It causes reduction of leaf mesophyll conductance, reduction of leaf hydraulic conductance. It causes leaf senescence. Okay, it controls the leaf expansion also. It inhibits shoot growth. Okay, in the root, it helps in root elongation, lateral root for it inhibits the lateral root formation, so only the tap root system will grow. And these are some of the important functions of abscisic acid, which is a stress hormone. Okay. Next, we are going to move on to the photoperiodism, which is uh, the physiological reaction of the organism, basically the plants, to the length of night or a dark period. Okay, you can also say to the length of the to the length of day. Okay, it occurs in both plants and animals because there are some nocturnal animals also which are active during the night, diurnal and nocturnal animals. Okay, but here solely we are going to focus on uh, the growth that is shown in the plants, which depends on the length of the day. Okay, it can be defined as the developmental response of the plant to the relative length of light in dark periods. So on the basis of the length of the day, okay, we have three types of plants: short day plants, okay, like tobacco and soybean. Short day plants are those plants which require 12 less than 12 hours of light for flowering. Okay, there are long day plants also. Long day plant requires more than 12 hours of light for flowering, like radish, spinach and oat. Okay, there are some plants which require equal length of day and night for flowering. They are known as day neutral plants, like tomato, cucumber, maize, etc. This can be explained properly with the help of a picture, okay, where you can see in the long day plants, okay, uh, this is the time of the light from 6 a.m. to 6 p.m. Okay, and this is like for 12 hours the light is given to the plant. Okay, you can see the short day plants have not grown, but the long day plants have, they are showing the growth. Okay, now in the second example, from 6 p.m. to 6 a.m., the light is, uh, the dark period is of around 12 hours. Okay, And you can see the long day plants have not flowered, but the short day plants have flowered. When they equal, okay, in this particular example, it is explained that if during the dark period, even if there is a brief flash of light given, okay, the long day plants will not get affected. Okay, sorry, the long day plants and the short day plants both are affected. How they are affected? The short day plants will not flower if their dark period is interrupted with even with even a brief flash of light. But this brief flash of light that is given during the dark period will help in promoting flowering in long day plants. So this explains how sensitive the short day plants are as compared to the long day plants. If during the light period of that is given to the long day plants, it is interrupted with a short dark period, then the long day plants will not get affected, they will still flower. Okay, but if the dark period of the short day plants is interrupted with even a brief flash of light, the short day plants will not flower. So they are more sensitive than the long day plants. Okay, let's move on to the concept of receptors. Okay, this is the basic pathway of a nerve impulse. Okay, that is described by a stimulus response model. So a stimulus is any change that is taking place in the environment, and this change in the external or the internal environment is detected by a receptor. Okay, receptors, they help in carrying the nerve impulse uh, to the to the sensory to the central nervous system, okay, with the help of sensory neurons. Okay, so when the response is selected by the central nervous system, then the signal is transmitted via neurons, that are the motor neurons, to the effectors. Okay, effector organs can be either muscles or gland. They are responsible for giving a response to a particular stimulus, and response is a change in the organism resulting from the detection of a stimulus. Okay, so this is the structure of a nerve cell. Nerve cell consists of the following important parts. There is a cell body which consists of a nucleus, okay, and all the other organelles like endoplasmic reticulum, mitochondrion, ribosomes, etc. The processes that come out of the cell body are known as dendrites, okay, and out of all the dendrites, one of them is very long, and that is known as the axon. Axon ends into axon terminals, which are known as telodendria. Okay, and the synaptic terminals is the junction between the axon of one neuron, okay, and axon terminals of one neuron and the dendrons of the next neuron. So this particular neuron is uh, non-myelinated. As you can see, it does not have any myelin sheath. Okay, but this second picture shows the myelinated neuron, where the myelin sheath is covering the axon. Okay, so cell body is basically the cell's life support center. Without cell body, the neuron will not work. As I said, there is nucleus and other important organelles present in it. Dendrites are the processes which receive messages either from the other cells or from the other neurons, obviously. Then from the dendrite, the nerve impulse then travels to the cell body, and from cell body, it moves towards the axon, which passes messages away from the cell body to other neurons or to the effector organs like the muscles or the glands. Okay, direction of nerve impulse also is shown through this arrow, as you can see. Then the nerve impulse moves to the terminal branches of the axon, with junction with other cells, and myelin sheath helps speed up the neural impulses, because you can see there are some uh, places where the myelin sheath is not present. Okay, the this region on the axon where myelin sheath is not present, it is known as nodes of Ranvier, and these nodes are the places where the nerve impulse jumps, Okay, and that is how it helps in speeding up the process of transmission of neural impulse. Okay, so here I'm just going to list down the important parts of the neuron, which is cell body, okay, dendrites, and axon. Okay, axon is covered by myelin sheath. Okay, the regions where myelin sheath is not present is known as nodes of Ranvier. Okay, and there are axon terminals also. Okay, in the nervous system, apart from nerve cells, there are second type of cells which are glial cells. Now glial cells are not involved in the transmission of the nerve impulses. They are present everywhere in the nervous system, and they act as supporting cells. They help in maintaining the structure of the neurons, firmness of the neurons, and they just support them in their structure and in their function. So these glial cells, they are present mostly around the cell bodies and the axons of the neurons. And if we talk about the types of glial cells, there are oligodendrocytes, okay, which are present in the, these are the glial cells of the CNS, okay. And there are second type of cells, the glial cells which are present in the peripheral nervous system, okay. These glial cells are known as Schwann cells. So they are present in the peripheral nervous system. Okay, Schwann cells help in producing myelin sheath around the axon. Okay, so yes, this picture shows the stimulus response pathway where the stimulus is a glass of water. It is seen by our eyes, which is the receptor. Okay, this stimulus is sent via the sensory neurons to the brain. The brain interprets the stimulus, and this interpreted stimulus is then moved via the motor neuron to the effector organ, which is our hand, okay, muscles of the hand basically. And the response is picking up the glass of water. Okay, so this is a simple example. Now let's move on to the questions. The cell that transfers information about pain to the brain is called neuron. Okay, now if you talk about a type of neuron which does this, it is the sensory neuron specifically. Okay, it is carrying the signal from skin to the brain, the pain signal. Okay, next question, we have to complete the following table. So first column is for the hormone, second column is for the location. So for the first one, the location is shoot tips and it causes cell elongation. So this is definitely auxin. Okay, second column is for fruits and seeds. It is found in fruits and seeds, but what it is doing in fruits and seeds? So this one is cell division. Okay, because see, in fruits the ripening can take place, okay, but we cannot talk about ripening in the seeds. So cell division is taking place, and that can take place by cytokinin. Okay, so auxin, cytokinin are the two important hormones. Then Anand went to the fruit market along with his father to buy fruits for the function. He saw the fruit dealer putting small quantity of some powder wrapped in a paper in each wooden pack containing unripe mangoes. On inquiry, the fruit dealer told him that the powder is a specific chemical which will help the mangoes to ripe early. Saransh was not convinced, and he discussed the incidents with his class teacher the next day. On the basis of the above passage, answer the following questions. Name the chemical that fruit dealer had kept in wrapped paper in each box. Is this practice healthy? Give your comments. So we are going to answer here. For the first answer, the chemical that the fruit dealer was putting wrapped in paper, okay, is ethapone, and it is a chemical, uh, artificial form of ethylene. Okay, artificial of ethylene. This practice is definitely not healthy, okay, because it is an artificial form of ethylene and it can cause side effects in the consumers. Okay, let's see what is the second question. Name at least one other plant hormone which promotes fruit growth and which promotes the falling of leaves. So, the hormone that promotes fruit growth is gibberellin, okay, and which promotes falling of leaf, the hormone which promotes falling of leaves will be abscisic acid. Okay. Then what values were shown by Anand? Anand was well aware about the harmful effects of the chemicals. Harmful effects of the chemical, and he was curious also. Okay. Let's move on to the next question. State the main function of abscisic acid in plants. Abscisic acid, I'm writing the abbreviated form, that is ABA. Abscisic acid causes abscission of uh, parts of the plant like leaves, fruits, okay, and it also causes seed dormancy. It also promotes the dormancy of the seeds. Okay, so let's move on. How do auxins promote the growth of a tendril around a support? So, as you already know that auxins, they induce cell division and cell elongation. Okay, so due to this property, when there is a touch stimulus received by the tendril, auxins they move to that part of the from the tip of the tendrils. Okay, they promote cell division and cell elongation, which causes the growth of tendril around the support. Next is, how is the movement of leaves of a sensitive plant different from a movement of shoot from light? So the movement of shoot from the light is due to it is a growth movement first of all. Okay, movement of shoot due to, okay, movement of shoot due to light is a growth movement. Okay, and okay, this basically I have to write here. Yeah, so movement of shoot due to light, it is a growth movement. Okay, and secondly, it is the movement towards the direction of the stimulus, but the movement of a sensitive plant, okay, it is not a growth movement, okay, and the movement also is not in the direction, it is not necessary that it should be in the direction of the stimulus, okay, like mimosa pudica. The next question is, if a ripened fruit is kept in a basket of raw fruits, what will happen? What causes it? If a ripened fruit is kept in a basket of raw fruits, the raw fruits also will get ripen due to the release of ethylene by the ripened fruit. Okay, so during ripening, fruits release a lot of ethylene, and when such a fruit is kept with the raw fruits in a basket, then it will cause the ripening of other fruits also. Okay, then okay, which of the following plant hormones is correctly matched with its function? Abscisic acid promotes seed dormancy. Okay, gibberellic acid promotes fruit ripening. Auxin promotes leaf senescence. Cytokinin promotes seed dormancy. Uh, Auxin and cytokinin, they do not promote leaf senescence and seed dormancy. Okay, they will help in, they are not stress hormones basically. And gibberellic acid, it does not promote fruit ripening, okay, it can promote fruit development basically, but abscisic acid here uh, pro- promotes seed dormancy, which is the correct function. Okay, next is the plant growth regulator involved in aging and ripening of fruits is, so this one is the easiest one. Ethylene helps in ripening of fruit and it also helps in senescence of the plants. Next question is, which of the following hormones is not found in plants? GA is for gibberellic acid, ABA is for abscisic acid, IAA is for indole acetic acid, which is a type of auxin. Okay, it is a type of auxin. 2,4-D, this is a natural auxin basically, which is found in the plants, but 2,4-D, which is 2,4-dichlorophenoxyacetic acid, it is a type of auxin, but it is artificially synthesized phenoxyacetic acid. Okay, and it is used as a herbicide basically. So this is an artificial auxin and it is not found in plants and it is used as herbicide. So that's all. Stay tuned for my next video. If you have any doubts from this video, you can post them on the forum and I will be happy to answer them. See you in the next video.